suppose I should preface that what I'm going to try to do this morning is to give an overview as to these sort of techniques. So for those of you that are very familiar with analysis of the gut microbiota, it might seem a bit patronizing, but hopefully you bear with me. And this ideally will set the scene for the remainder of the talks during the course of the day. And as a Corkonian, I should also say welcome to all of you, and I hope you enjoy your stay. So what I'm going to try to do over the next 25-30 minutes or so is to give an overview into different culture-based approaches initially, because those were the traditional and classical ways of studying microbiology. Then the newer sequencing-based approaches that we're using increasingly as time has gone forward, introduce some of the terminology and concepts, and then discuss some specific issues such as where you might want to be collecting samples from, issues relating to which technology you use, and then finally just some example of the applications that we are involved in. So within Teagasc, within the APC, we do have a platform that focuses on characterizing the microbiology. I lead the DNA sequencing center, and the sort of things that we study there involve looking at the gut microbiota and how it differs from healthy versus disease, from people who are, for example, athletes versus non-athletes, and the impact of exercise on the microbiota. And my colleague Orla is going to talk about that in the next presentation. But we're interested in comparing people who are lean versus obese, the microbiome, in other words, the fungal component of the microbiome, mother-infant relationships, the vaginal microbiome, and a broad variety of other things also. At our centre, we carry out DNA sequencing on behalf of researchers at the APC and Teagasc, but we are also engaged in quite a number of collaborations. We're not designed really to make money, but rather to carry out good science. So we also look at a variety of other different microbial communities, such as those in the soil, the pig gut, the cow rumen, poultry microbiome, and a broad variety of other locations within the human body also. So in terms of the stats and figures, you will have, if you've attended any microbiome conference in recent years, you'll know very many of these. But I suppose the fact that has become very much apparent in recent years that there are microbes right throughout our human body, on our skin surface, in oral cavities, on our lung. In breast milk, there's a suggestion that there are microbes in placenta, somewhat controversial. But even just looking at the number of publications that have resulted in recent years, you get a real sense as to how much that field has exploded. The gut, colon, small intestine area has really been a focus of a great deal of attention, and you will see that of a total of 17,000 papers in that category, almost 11,000 of those have appeared in the last 5 years, up to 2016, and that's obviously after increasing substantially since then. Other areas of slightly lesser interest include the oral, mouth, tongue cavity, which is 2,000 publications, urogenital vaginal area, skin, airway, lung, down to even the eye microbiome in recent times. With regards to the microbiome, I suppose one of the big feature of it is that it can be changed, and it's constantly changing. If you sequence your human genome once, you have that on record, and that's not going to change for the rest of your life. The microbiome does have the potential to change, and in fact changes quite dramatically at different stages during our life, especially when we're infants, as weaning occurs after exposure to antibiotics, and then again changes substantially later on in life as well. There are things that you can do to change your microbiome for the better and for the worse, and that can have a broad variety of different impacts on your health, including your mental well-being. And so the things that you can use in order to change your microbiota for the better or worse includes things like diet, probiotics, prebiotics, antibiotics. And maybe just to expand on it here a little bit, some of the things that we do to try to determine what the impact or how to change the microbiota and its impact on health are to carry out a broad variety of different human studies. Studies with animals, different type of animal models, mice and pigs. An interesting type of model that people use frequently when studying the microbiota is to look at germ-free animals, so animals that have no microbiome at all, and you can add in individual microbes or clusters of microbes to determine their impact. And also we have a broad variety of different in vitro and ex vivo models whereby we can transplant a microbial community from a gut or from a fecal sample into a fermenter, and then determine what the impact of that gut microbial community is as a consequence of a broad variety of different interventions. And just to return to those interventions again, so these may be classical antibiotics, or maybe bacteriocins that are produced by a probiotic, different prebiotics which are designed to target particular components of the gut microbiota and encourage them to increase in number, fecal transplant, which I believe we'll be returning to in a later talk this morning, diet, and also exercise. And the reason for all of that, and this is only a quick overview of the sort of things whereby the microbiota can contribute to health, but they relate to things like vitamin synthesis, digestion and absorption, immune stimulation, the control of pathogens either through the production of antimicrobials or through the attachment of microbes to the gut and preventing a pathogen from attaching and causing disease, influencing the efficacy of energy harvest, so that has implications with regards to obesity, but also people who don't have sufficient nutrients in their diet, intestinal epithelial cell proliferation, signaling from the periphery to the brain, and you could speak for 30 minutes on this topic alone. So getting more into the microbiology side of things, so why has there been an explosion in this whole field in recent years? And there really has been as a consequence of the development of culture-independent approaches to microbiology. And these developments do not have influences solely in this specific area, but to every field of microbiology, regardless of whether somebody is studying the soil microbiome, or the microbiome of tundra, or of a desert, or, as I said earlier, of the rumen of cattle. So traditional approaches involve actually growing the microbes on agar plates and in broths and different kind of soupy type materials. But in order to really get a good appreciation of all of the microbes in your gut, one would need to have an agar plate that grows every type of microorganism that's present, and that just isn't practical. First of all, very many of the microbes present are anaerobic. They don't like oxygen. Some are microaerophilic. They like small amounts of oxygen. Some need bile. Some don't like bile. Some need specific levels of salt. Some don't like salt. So you would need to, first of all, generate 50 or 60 or maybe several hundred different types of agar plate in order to grow those microbes in the first place. And then incubate each of those at a different temperature and in different levels of oxygen. So no such culture conditions exist, and even then there are some microbes that we know are present. We know from sequencing-based approaches that they are present, but we haven't yet been successful in growing them. Hence the need for culture-independent types of approaches. So DNA sequencing-based approaches allow us to get a full insight as to which microorganisms are present in that community. And I'll be referring quite a lot to metagenomic DNA later, so that's the terminology used to describe all of the DNA that's present in a microbial community, which is then subsequently analysed. Now, before I move away from classical approaches, I should explain that microbiologists haven't given up on these yet, and there have been new initiatives to try to address those issues. So culture-based approaches can and have been improved as a consequence of using anaerobic hoods, using newly developed culture media with special ingredients to encourage the growth of certain microbes. You can use heme or blood products in order to encourage the growth of certain microbes as well, rumen fluid. Some microbes don't grow well on their own, but if you use a co-culture type approach whereby you have another microbe present with it, that facilitates its growth. There are systems like the iChip-based system, which was developed for soil-based systems, whereby the different approaches were used to try to isolate microorganisms in a chip, but then to reintroduce that chip into its natural environment by reinserting it into a soil. The same principle holds true for introducing such chips back into fecal material, whereby the other microbes can't get access to the inside of the chip, but the nutrients that are there can transfer across and the microbe can grow successfully. The growth of acidophiles, so microbes that like low pH, can be encouraged by using specific acids. Halophiles like salt. Microcolonies, or colonies that in the past people would ignore because they're tiny little pinprick colonies that grow adjacent to their healthy colonies on an agar plate and would have been ignored previously. But now it's appreciated that some of these are unusual species that are worth closer attention. And then finally, because of the advanced insights that we're gaining through culture-independent approaches and DNA sequencing, we now know lots about microbes that we've never grown previously. And we know which metabolic pathways are present in those microbes. And that knowledge can now be harnessed in order to make completely new agar-based media to facilitate their growth now. And then finally, something that's close to our own heart in recent times has been to target other components of the gut microbiome. So there is a big emphasis on bacteria, but people in the APC and elsewhere are looking at the phage component, the viral component, and we're doing quite a bit of work with the fungal component, or that which is known as the microbiome. I mentioned that there's a broad variety of different approaches that one can take with different agar-based strategies, and this is an example from just one paper that whereby they claim to use the approach known as Culturomics. And in that paper, after playing around with hundreds of different culture media, they came up with a list of the 70 most effective culture media. And this is just the first 19 of those, but it gives you a sense as to how many different variables you can change in order to grow these microbes. Once you grow the microbes, then there's a broad variety of different things that you can do with them. If you're dealing with microbes on a large scale, then typically you would use some sort of robotics-based system in order to transfer the colonies into 96 or 384 well plates, and then use high-throughput screening on agar plates or other broth-based systems with cell lines in order to assess what these microbes are capable of doing. So this is the sort of thing that we do when we're looking for new antimicrobial producers that could potentially be probiotics that could change the gut microbiota. So you can see a broad variety of different zones of clearing of different size, representing different microbes producing different antimicrobials. And then the principle would be to identify one of these microbes, carry out perhaps genome sequencing of the strain to identify which gene clusters are contributing to this phenotype, and then ultimately identifying the specific gene cluster responsible. So that leads us nicely into DNA sequencing. So this is one obvious application of DNA sequencing. In this instance, we're using a culture-based approach, so we can grow the microorganism. We genome sequence the microorganism, and we figure out why it has a certain activity. Genome sequencing is also relevant to culture-independent approaches, in that in recent times that's become possible without ever growing the microorganism. In the past, it involved picking out information that's present in the large metagenomic DNA and trying to put it together to make sense as to which bits belong to which individual strain. But now through newer approaches, it's now possible to carry out genome sequencing on single cells. And the principle there is to isolate the individual environmental microbes and to separate them and encapsulate them in such a way that one individual microbe ends up in one particular capsule. You can then introduce the various different requirements for DNA sequencing, the various different enzymes and nutrients which pass through the capsules by diffusion. And by fragmentation and subsequent sequencing, you can sequence each of these different cells in isolation and then put together its genome sequence subsequently. And these are the sorts of instruments that you would use for this principle, and I'll come back to some of these later. So Aaron from our lab prepared this review a number of years back and prepared this image for the review. And what we're also highlighting here is what you can do with regards to culture-independent approaches, but looking at entire communities. And obviously in this context, it relates to the gut. So your options are to look at metagenomic DNA, so all of the DNA from that particular environment, and to study that, or to isolate all of the messenger RNA from the environment and to determine what genes are being transcribed by the microbes in the gut. So this would be known as metatranscriptomics or RNA-seq, and it gives an insight as to microbial gene expression. I'm going to focus on metagenomic DNA because that's applied more frequently. And in that situation, there are normally two different strategies employed. One whereby you focus on a marker gene, such as the 16S gene that's present in bacteria, or the ITS gene, or the ITS region, which is present in fungi, or else you carry out whole metagenome shotgun sequencing. And I'll delve into those in a little bit more depth in a moment. I said I'd come back to the sequencing platforms, and I don't expect you to read all of this information, but you can look at this site if you would like to look at it more closely. But I suppose the point I want to get across simply here is that there are different types of sequencing instruments, and it really is a case of horses for courses. So which DNA sequencer you want to employ depends ultimately on what you want to do with it. So Illumina are the market leaders. They produce 80 to 90% of all sequencing that's carried out is with their own instruments. You might be familiar with the MiSeq, the NextSeq, and the HiSeq. And its closest competitor would be the ion torrent technologies, ion PGM, ion proton. And the principle underlying these sort of technologies is that they generate short DNA reads, but generate very many millions of them. So it's a numbers game. Alternatively, you can look at sequencers that generate longer reads, such as those supplied by PacBio or Oxford Nanopore, which generate longer reads, but for now, not quite so many of them. And longer reads are beneficial for certain applications, but if you have samples that you want to really analyze in a huge amount of depth, then the numbers become more important than the read length. So ultimately, it depends on what you want to do. In terms of how this whole process has evolved, at our DNA sequencing facility, we got our first sequencer around 2008. We thought it was fantastic and we were really at the state of the art. We could generate 1 million DNA sequence reads with our Roche 454, and one run costs in the region of 9,000 to 10,000 euro. Subsequently, we began to use that less and less as new sequencers became available. We have an ion PGM, which will generate about 4 million DNA sequence reads and is useful for whole genome analysis and looking for the composition present in microbial communities. We use our suite of MiSeq instruments to carry out whole genome sequencing, some compositional analysis, in other words, identifying the names of the microbe in a community, as well as shotgun, and I'll explain in greater depth the difference between the two of those now. But if you even just think that between this instrument and that, we can now generate 15 times more data at about a quarter of the cost. So it might come as no surprise that a year or two back we threw this out in the rubbish heap, despite having spent hundreds of thousands on it when we first purchased it a number of years back. We have other instruments that provide even more DNA sequence reads, such as the ion proton, up to 30 million reads. Or the highest output instrument that we possess is a NextSeq, which generates up to 400 million reads now. And the larger equivalent of that, the HiSeq, is also available. Something that's attracting quite a bit of interest in recent times relates to the Oxford MinION. It is quite an interesting instrument. In size, it's about the size of the pointer here. The principle is that in theory you can carry out DNA sequencing anywhere with it. Now you obviously have to be able to extract the DNA and to prepare it in advance, but it does provide real-time DNA sequencing information. The strategy used by that company is quite clever, so they put a lot of money into marketing, but their R&D facility, to my mind, it could do a bit more work, but they overcome that by providing these DNA sequencers to people with expertise in the area and allow us to play with them and to optimize them and ultimately improve the product. But they do have newer versions of that instrument that generate even greater amount of DNA sequence reads as well, and they're likely to be a major competitor for Illumina in the future. Okay, so in theory, with some of these new benchtop sequencers, everybody could have a sequencer, but I don't think it's really quite as simple as that, because there's a number of different things to consider. First, you need to have instrumentation and expertise with regards to how best to extract the DNA, to prepare the associated libraries, and to make sure you don't get cross-contamination. You need to be able to quantify the DNA, ensure its quality, and then have various different means of storing the data, and analysis is a key feature. So initially, for quite a number of years, people came to us because we had expertise in being able to generate DNA sequence data, but increasingly people are coming to us now because we can also help them in doing the analysis. One can get commercial providers to do analysis for you, but you just need to be a little bit careful about this kind of plug-and-play type approach, because quite often the way in which you carry out an analysis will give you completely different results. So you really should have good oversight or good appreciation as to why a particular strategy is being used. So returning to this metagenomic sequencing and the differences between the two of them, I'll go through it in a bit more depth here. So ultimately, if you are looking at community DNA, regardless of what you need to do with it downstream, the first important thing is to be able to extract all of the DNA from the community and to ensure that the strategy you take isn't selective and results in you missing out on some communities that are present. So compositional sequencing is probably the approach that people are most familiar with and is used most widely. You might be more familiar with the term 16S sequencing, but this is only compositional sequencing as it's applied to bacteria and not fungi or viruses. So the 16S sequencing approach relies on the fact the 16S gene is present in all bacteria. It has some useful features in that there are conserved regions at either end of the gene, but the region between those conserved bits are very variable. So you can take advantage of the conserved regions to carry out a PCR amplification, and then by sequencing the regions between that, you can identify different variations, compare that with databases, and determine just what exactly what's present and how that corresponds with microbes that are already in the database to ultimately identify the microbes which are present in your community. And you get an output that's a percentage. So 10% of the total microbial community present is a lactobacillus, or 5% is a bifidobacterium. Now, that sort of technology really can only reliably give you information down to the genus level. For some types of microbes, you can delve a little bit deeper to the species level, but we normally just stop at the genus level. So there's somewhat of a limitation surrounding this approach. The other limitation is that you only get information about the identity of the microorganisms, and for 16S, it's only telling you about bacteria. Shotgun sequencing is more expensive, but it's the approach that we use in all instances where possible at the moment. And it relies on the fact that instead of just focusing on one gene, you're sequencing as many of the different genes that are present in the community as possible. And so you're really sequencing in a willy-nilly, random type manner, but as a consequence of that, you're getting information about lots of different genes that are present. So genes involved in metabolism, antibiotic resistance, you're getting an insight into the phage community, the fungal community, the archaea, the larger eukaryotic microorganisms that are there, as well as getting an insight into the identity of those, and importantly, you can get information down to the species level as well. So, to my mind, it gives you everything that 16S sequencing can do, plus an awful lot more. So some other things to consider. What biological material are you collecting? Are you focusing on fecal material or some material from elsewhere in the intestine? How should it be stored? What DNA extraction protocols should be employed? What DNA sequencing platforms should be used? And what bioinformatic pipelines should be used? And all of these are of key importance. And if you change one of these, it can very dramatically impact on your ultimate final outcome. So again, something that you need to be aware of if you're entering into this field for the first time, and you really should get advice as to how best to proceed rather than continuing without being really sure what you're up to. In terms of 16S analysis, so just to highlight the importance of some of these different features, so we did a study a couple of years back with Marcus Claassen, who's going to be speaking later this morning. And we looked at the impact of different extraction protocols. For the PCR primers, for 16S, you can actually use different primers because there are different regions within the 16S that you could, in theory, sequence. And then looked at different instruments. And so we got a mock community, so we kind of made up our own community of pretend gut microbes where we got the DNA from these and mixed them up. So we kind of knew what the answer should be to our sequencing run before we did the sequencing. And then saw that out of 21 different species that we knew were present, these were the number of species that we actually detected. So depending on which type of primers we used, which instrument we used, we got different outcomes. So rather than identifying our 21 species, sometimes we only got 11 of those being detected, other times maybe as many as 20. So again, these are the sort of outputs that you get from your analysis, and what I'm showing here are the different types of sequencers, an ion sequencer versus a MiSeq sequencer, and different PCR primers. So you're seeing across the different sequencers, sample number 1 versus sample number 1 here on the different sequencer. It's somewhat similar when the same primers are used, so that's reassuring. But then if you compare the different primers, within the same sequencing technology, you will get a completely different output. So ultimately, what's the bottom line? I think that care needs to be taken when you're standardizing protocols within your lab in terms of treating the samples, how you extract them, which polymerase you use, which PCR primers. And especially care needs to be taken if you're trying to compare your results with those from another lab, because the differences you're seeing between your study and someone else's study may not be due to biological differences, but may be due to the strategy that you employ. We've also taken a similar approach to determine how these different variables impact on shotgun sequencing. In this case, we picked a simple microbial community from kefir. Kefir is a fermented milk, which has a low diversity community. Now, as you can see, we're getting down to species level detail here, and across each of these 6 samples just simply represents a different kefir sample, because each of these fermented milks is different. What you're seeing as you're going down are the results from a MiSeq instrument versus a NextSeq instrument versus a proton. And then what you're seeing here are Kaiju, Kraken, and Metaflan. And these are different systems which are used for classifying bacteria to, in other words, take your sequencing, your shotgun sequencing data, and to determine what are the names of the bacteria which are present there. So you'll see that sample number 1 on the MiSeq, sample number 1 on the NextSeq, and the proton have some differences. 2 is quite similar to 2 to 2, 3 is quite similar to 3 to 3. So that's somewhat reassuring in terms of at least shotgun sequencing and the different sequencers. But then if you look within a particular instrument, you can, you're seeing things differ when you're looking at different classifiers. So depending on whether you use Kaiju software, Kraken, or Metaflan, you're seeing other populations here which are being identified by Metaflan, which are not present at all when you use these other types of software. To emphasize that more dramatically, again, we went back to our model pretend communities and made those up again. And you can see there are similarities when you're looking within the same software across different instruments, but when you're looking at different software, you're seeing considerable changes in terms of the proportions of different communities which are present. So the net outcome, the net take-home point, you're seeing somewhat consistent results when different sequencing platforms are used, but different when different bioinformatic tools are employed. You can, going back to the overall purpose of the presentation and just talking about characterization of the gut microbiota, quite often once you generate this data, you want to compare it with different forms of metadata to see what sort of correlations are present. You can also determine what sort of impacts the gut microbiota are having by looking at the metabolites which are being produced by those gut microbes within the gut, so looking at the fecal metabolome, or determine the impact it's having on the host by maybe, for example, looking at the metabolome of urine collected from individuals. Now, in addition to looking at the identity of the microorganisms which are present within a community, one of the other outputs that people are particularly interested in is to look at diversity. And so sometimes this is confusing for people who aren't familiar with the background to this. But these are, in essence, terms that we've borrowed from ecologists. And so an ecologist, when they're talking about alpha diversity, are referring to how many different types of sequences, or in their case, how many different types of species are present in an environment. So if you're looking at the Amazon rainforest, you're trying to see how many different animals or insects or plants are present within a particular region. So that applies also for gut microbial analysis. For beta diversity, it's a different focus. So instead of looking at one individual community, what you're doing is comparing 2 different communities. So the extreme examples here would be a high diversity community of the Amazon versus a desert low diversity community. So when you apply those terminologies to the gut, it's become apparent through a vast variety of different studies that low total diversity is undesirable in a vast majority of cases. And so you see lower diversity in scenarios where people are suffering from infections such as C. diff. Individuals with obesity typically have lower diversity. IBD and IBS also have lower diversity in general. And then some other individuals have unusually high microbial diversity, and we found that and were surprised to see it when we looked at a group of elite professional athletes in a study a couple of years back, and I think Orla is going to return to this later on. Again, when you think about diversity, what sort of factors impact on your output there? So going back to our different kefir samples, what you can see is we have different DNA sequencing instruments. This sort of yellow color for the MiSeq, red for NextSeq, and black for proton. For those of you not familiar at looking at these sort of MDS plots, in essence what you're doing is looking at one spot that represents the entire community, which is capturing all of the information about that microbial community. And when a spot is located, or a square is located close to another square, it means that the community is quite similar in terms of its bit diversity. So what you're seeing is that all of the arrows and circles are over here. So all of the sequence analysis that's being put through the software known as Kraken or Kaiju is giving you a similar output. But if you look at the exact same data using a different classifier, you're getting quite a different output and a very dramatic consequence of that. Then just finally, so I mentioned that there's a lot of different factors that can also impact on your output, such as how you store your DNA, whether you extract DNA from it when it's fresh or frozen, and there isn't time to delve into all of that just now. But we were interested in an interesting study a number of years back where we looked at UC and controls, and we're lucky enough to work with a clinician who was able to access samples from different locations within the large intestine to collect luminal samples, whole mucosal biopsies, and also the mucus gel there. And from different regions from a longitudinal perspective as well. And then to sequence those. And I'm not going to get into the results in terms of what happened from a UC versus a healthy approach, but what's very much apparent is that when you compare a lumen sample versus a mucus sample, the levels of particular microbes which are probably having an important role are very much different. And so if you were to only look at the lumen sample, which is more representative of what you'd find in a fecal sample, that output will be quite different from mucus. But maybe it's the microbes which are present in the mucus which are having the greater impact in terms of the immune system. So those are all considerations as well. So I'm conscious of not lingering too long, so I'm going to leave it at that. By way of summary, I suppose I'm hoping that I got across the following points. In other words, that characterization of the gut microbiota can still involve culture-independent approaches as well as culture-independent approaches. When you're employing DNA sequencing, that can be done in a variety of different ways through genomics, metagenomics, or metatranscriptomics. A lot of people are doing metagenomics, and that involves amplicon sequencing and increasingly shotgun-based approaches. Which you use depends on, to some extent, your budget, but also what questions you want answered. Outputs typically focus on the assignment of taxonomy, functional potential, or determination of diversity. But if I've gotten you to leave today with just one particular point, I hope that point is that the outputs from those analyses depend very much on quite a number of different factors, and those include the bioinformatic pipelines used, to a certain extent the sequencing platform used, sampling site, and a number of other things like DNA extraction kits, storage of samples, and so on. And finally, thanks to all of you for your attention, as well as to all of these great people who did the work.